Tasha is our first speaker. She's an associate professor in epidemiology and in biostatistics. She co-directs the master's and PhD program in epidemiology, and her work is in genetics of diabetes and lung disease, and I believe she's going to talk about lung disease today. But I'd like to show that this demonstrates the very strong collaboration that our department has with the School of Medicine and Department of Medicine in areas of research.
Good morning, everyone, and thank you, Jill. I'm very happy to be able to represent the department today and share with you some of the most recent findings that we have in our studies of pulmonary fibrosis. Idiopathic pulmonary fibrosis is a chronic disease of the lung that's defined by progressive scarring or fibrosing in the alveolar interstitium, that is, the areas around the air sacs in the lung that are very important for maintaining the structure and function of those air sacs. It's a disease that occurs mostly in older adults. The age of onset is typically between 50 and 70 years of age. And in the U.S., it's been estimated that there are 38,000 new cases diagnosed each year, although there's some interesting research to indicate that maybe both the incidence and prevalence are increasing. The disease progression is characterized by worsening shortness of breath and lung function over time, and the diagnosis of IPF is devastating. The median survival is only 3 to 5 years after diagnosis. There are no approved pharmacologic treatments in the United States, and lung transplant is the only intervention that's known to prolong life. The etiology and mechanisms of disease for IPF are largely unknown, although there are some causes that are beginning to emerge. It's a complex disorder where there are likely multiple genetic and environmental risk factors that are interacting to influence risk of disease. The strongest known environmental risk factor is cigarette smoking, although almost 40% of individuals with a diagnosis of IPF have no personal history of cigarette smoking. The most strongly associated genetic factor that had been identified that has been identified is a common genetic change in a mucin gene that creates mucus called the MUC5B gene that significantly increases the risk for IPF. And this common change was identified by Dr. Max Seibold, who's now a faculty member at National Jewish Health, and he identified it when he was a postdoctoral fellow in the lab of my very close collaborator, David Schwartz, who's in the Department of Medicine here. We reported more recently, earlier this year, based on several thousand cases and controls, a very strong effect of having either 1 or 2 copies of this T allele, which is the allele that predisposes to disease, compared to having no copies of those alleles. And because of the very large sample size and the very large effects, there was a very striking level of statistical significance for this finding. So with a risk factor this strong, one of the questions becomes, what else can we learn about disease based on something that is this strong of a risk factor? And so the first study that I'd like to give you a little more detail on is a study that was led by our outstanding postdoctoral fellow in epidemiology, Dr. Anna Pelto. And she examined the study, or examined survival in IPF related to this genetic variant that is a very strong risk factor. We considered 2 different cohorts for this study, one that I'll call Nspire and one that is Chicago. The Nspire cohort consisted of individuals that were recruited for a clinical trial by the pharmaceutical company Intermune. And the Chicago cohort was recruited as patients that were seen in an interstitial lung disease clinic at the University of Chicago and were collected by our colleague, Dr. Imre Noth. We had a larger sample in the clinical trial sample than we did in Chicago. The Nspire cohort was initially designed to be sort of the derivation cohort, and then we would test to see whether things were actually relevant in another cohort, the Chicago cohort. You can see that they are similar in terms of the sex distribution. But by design, the Nspire cohort was collected to be at an earlier stage of disease, and so they were also younger at age of onset and had better lung function, compared to the more moderate stage of disease that represented the Chicago cohort. And the frequency of the risk allele was very similar between the 2 cohorts, and very similar to what we've seen in a broad range of populations. If we first look at just the very simple Kaplan-Meier estimates of survival over days of follow-up, you can see that individuals who have 2 copies of the non-risk allele actually have worse survival compared to individuals who have 1 or 2 copies of the allele that predisposes to disease. And we developed full Cox proportional hazards models to adjust for other important factors related to survival. And both age and other lung function variables were significantly associated with survival. But after adjustment, this protective effect in terms of survival of the risk allele was statistically significant. And we saw very similar results in the Chicago cohort, both in terms of just looking at the simple Kaplan-Meier curves and in terms of the adjusted hazard ratio estimates. Not all of the groups reached the median survival point in all of the different genotypic groups, but we can look at the survival time the rates or the time to 75% survival in the different genotypic groups, just to get a sense of what the magnitude of effect is of this genotype difference. And in this Chicago cohort, which again probably represents a more representative cohort of patients that you would see in a clinic, 25% of patients had died after 6 months, whereas it took 26 months for that proportion of patients to die in the group that had at least 1 copy of the risk allele, and then it took even longer for individuals with 2 copies. So while the T allele is a very strong risk factor for disease, it also appears to be defining, based on these data and other data that we have, to be defining a milder form of disease, if you could call any form of pulmonary fibrosis mild. And while this T allele is a strong risk factor for disease, it's present in 19% of the general population, and over almost 40% of individuals with disease do not carry any copies of the allele. So we hypothesized that there were other genetic variants that were probably acting either independently or in concert with the MUC5B gene to influence risk. And so we conducted a large genetic study to try to identify those additional risk factors. We conducted a 2-phase genome-wide association study, both a discovery phase and then we followed it with a replication phase. We included both IPF, which I've talked about, and other fibrotic interstitial lung diseases that have specific diagnoses, and then individuals who had definite fibrosis based on several criteria, but that did not fit any of the specific diagnoses. And we did this because there's substantial radiologic, clinic, and pathological overlap among these different diagnoses. In both groups, there were both, the majority of cases were what we consider sporadic. That is, there's no known family history for disease. And including, getting this number of cases required many collaborations with people across the U.S. and in Europe. We used 2 different out-of-study control groups to reduce the costs of this expensive study. In the discovery sample, we used over 4,500 controls that had been genotyped by our colleague Mark Lathrop at CEF in France as part of his other studies that were genotyped in the same fashion that our cases were genotyped. And all of the cases and controls were self-reported European ancestry, and in the discovery population, we were able to use the data that we had genome-wide to select a group of controls that were as well matched as we could get in terms of ancestry to avoid confounding by ancestry. And then we worked with Drs. James Crapo and John Hokinson in the COPD gene study to select an appropriate set of controls for our replication study. We searched for association across the entire genome, and what I am showing here are the locations of each of the places where we tested for association, starting with chromosome 1 all the way through the autosomes and chromosome X. And each dot on this plot represents a place where we tested for association. On the vertical axis is a transformation of the p-value, so that very small p-values are represented with a large value on this scale to make it easier to visualize. And this red line corresponds to an empirical evaluation for what reaches genome-wide statistical significance, accounting for all the tests that we did, which is a nominal p-value of 5 × 10⁻⁸. So now you understand why we have to have such large sample sizes. And what I'm showing on this graph is only the results for the discovery sample. And what we did is we selected all of the markers that were above this blue line, which was based on something more than this, but conceptually you can think of it as being bumping up above background in terms of potentially interesting. And we took that into the replication phase. And so the results that I'm going to be highlighting now, just using this simple graph, are those that after considering the replication data were genome-wide significant, even considering all the tests that we had done. And so as a very first good positive control, our strongest signal was the MUC5B gene that we knew existed and we knew had a very large effect. So that was comforting to see that result. And we also identified variants in 2 genes in the telomerase pathway where previously very rare variants, primarily in families, had been associated with disease. So these were very good positive controls. In addition to these, we identified 7 novel loci that I've highlighted in green here that are new genetic risk loci for pulmonary fibrosis. And in fact, in many of these regions, there are very specific genes where the association is very localized to these genes and does not appear to be spread out in a region that will require a lot of further work to narrow down. And these genes are what's been very exciting in terms of giving us new insights into what might be the primary defects that are actually leading to pulmonary fibrosis. And just as one example in terms of a new pathway, this DSP gene codes for desmoplakin, which is a very important component of the cell that has to do with cell-cell adhesion. So how well the cells are maintained in terms of their relationships for tissue integrity. Particularly in the epithelium, like the epithelia that line the lung air sacs in the lung. And similarly, DPP9 is related to cell-cell adhesion in epithelial tissues. And so others have shown that defects in these pathways in animal models can lead to disruption in the constitution of these epithelial tissues. And so one of our hypotheses is that individuals that may have defects in these pathways may be more susceptible to injury because they don't have tissue integrity in the epithelium. One of the really interesting things that we've also learned in this study is that the familial disease, represented in blue, and the sporadic disease appear to have similar effects for each of these loci. So I'm showing, if you look at the odds ratios and 95% confidence intervals across all of the loci that have been identified so far, you can see that the estimates are entirely overlapping between those who have a familial history and those who do not. And this has been very thought-provoking because one of the standard assumptions for many in the field has been that familial and sporadic disease are entirely different diseases that probably have different genetic underpinnings. Whereas these results suggest that, at least for these loci, the effects are very similar between the 2 different types of disease. And so that's inspiring us to look for other reasons why the familial disease might look different clinically from the sporadic disease. In aggregate, the biological functions of the new genes that we've identified have perhaps shifted us in terms of what we think the primary defects are in the pathogen that lead to pulmonary fibrosis. And what I have here is a very standard diagram that we put in lots of grants describing what the field thinks about the pathways that lead to pulmonary fibrosis, where there are a series of microscopic lung injuries that occur separated both in time and in space in the lung as a result of common stimuli like cigarette smoke and infections. And that really it's an aberrant wound healing process that goes awry that leads to interstitial lung disease. So there have been lots of studies that look at upregulation of repair mechanisms and things like that, and we see a lot of that in the disease. However, based on the results and the genes that we have so far, it also appears that we should probably be also focusing on the potential for both injury to happen and the severity of the injury, as opposed to necessarily having aberrant repair mechanisms. It may just be that there's lots of injury happening, and that's why all of the repair mechanisms are upregulated. I gave you the cell-cell adhesion molecule example, so where individuals may not just be able to accommodate the mechanical stretch of the lung or injurious particles. Another example is that MUC5B gene. It's a mucus-producing, it codes for a mucin, which is mucus in the lung. And one of the hypotheses for the mechanism is that that risk variant actually increases the production of that mucus. And so perhaps these injurious particles and other things are actually trapped longer in the lung and provide more injury than they would be if they were sort of efficiently cleared out. These are just hypotheses, and there's a lot we have to learn. And it certainly doesn't rule out the importance of wound healing, but this has created a big shift in the field in terms of thinking about alternative therapeutic targets that we could go after, because there are some targets that are actually already approved that might be for treatment in other diseases that may be relevant for pulmonary fibrosis. So as is typical, there are more questions raised than answered based on the research that we've done so far. One of the primary ones is, what is the molecular basis for the genetic associations that we've just discovered? So we have quite a lot of ongoing work looking at the mucin gene, but these other genes have provided other questions, both in terms of what's the actual genetic variants that's causing the risk, and then how is that actually leading to a difference in risk biologically. One of the things that some of our colleagues are very interested in is whether the genetic findings can point to new therapeutic targets, either existing or development of new ones, and whether the genetic variants can identify subgroups where treatments will be effective. There have been several failed trials. Most of them have collected blood and DNA, and so it would be a fairly simple thing to do a post hoc analysis and just see whether there might be evidence for subgroup effects. We're also interested in whether the other genetic changes influence prognosis, like MUC5B did. And one of the studies that I did not get a chance to go into more detail, but that we conducted in collaboration with our colleague, Dr. Matt Honninghake out of Boston, was that we identified or realized that the MUC5B variant is associated with both early interstitial lung abnormalities and definite fibrosis in a population-based cohort in the Framingham Heart Study. And this raised a lot of questions about both what the early abnormalities actually mean. A lot of them would have been dismissed, except for the fact that they are very strongly associated with this genetic variant that we know predisposes to clinical disease. And so there are a lot of questions about what those abnormalities mean, at what rates they progress to disease in the population, both in familial and sporadic groups. And so that's something that we're actually working on now in terms of grant application development to try to be able to study these things. So before I finish, I need to acknowledge the fact that I am simply representing a very large group of individuals who have worked on these studies, and I can't even represent them all on one slide. But primarily, I want to recognize David Schwartz, who is the PI of many of the studies that funded this work. I mentioned Dr. Anna Pelto, who's a postdoctoral fellow in epidemiology, who led the survival studies. And I need to acknowledge Dr. Weiming Zhang, who's a senior research instructor in the Department of Biostatistics and Informatics, who took the brunt of the quality control and data analyses for that large genetic studies, which was not a small undertaking. And then obviously lots of individuals that both contributed genotyping expertise and cases for the genetic study. And with that, I'd like to thank you very much for your attention. I'll be happy to take questions. Yes. Mm-hmm. It's not in the MHC region. It's quite far away. But there are, based on some new analysis that we've done that sort of takes advantage of some additional data we have, there does appear that there may be a signal in the MHC. That will be hard to tease out what that is, but the DSP signal is very independent of the MHC.
Tasha, this is very interesting. I wonder if at this point the group has thought about any treatment implications of any of these genetic targets.
Yes, thank you. That's a great question. We're actually in the process, David, Anna, and several colleagues are in the process of writing an application to look at whether or not there might be hope for some of the mucolytic drugs that are available, so reduce mucus production based on the MUC5B finding. And then we're also starting to work with other colleagues to try to do screens based on what these genetic findings are doing the screens for other compounds that are either in development or that we could try to develop that might target some of the pathways that have been implicated. So yes, that's the very next thing on the timeline, in addition to trying to understand what's going on preclinically to know if there might be a better time to intervene, since by the time we diagnose it, it's in a very bad state. Mm-hmm.
Any other questions?
So the similarity between the familial and the non-familial in the hits that you found in your GWAS, are there familial genes that you're just somehow not seeing? And if so, what sort of genetic epidemiology approach can you use for that?
Yeah, it's a great question. Yes, certainly. A lot of my colleagues want to say that the familial and sporadic disease are the same genetically, and we can't say that because we can only say what we've observed. So an important caveat to the genome-wide association study is that it only is powered to detect common variants, so things that are at an appreciable frequency in the population. What we're now doing is going back and we're doing sequencing across these regions in our families to identify, and actually more broadly, not just in these regions, to identify what we presume we will find are rarer variants that in these families probably increase the probability that familials will get disease. But we honestly don't know if that's the case. Is it a bunch of rarer things that are driving it, or is it simply that they have a higher burden of the things that are contributing to both familial and sporadic, or are there other environmental features? But we expect that we'll probably find the range of those things, actually. And so the approach that we have over time, we've been collecting a lot of families, and so now the approach is to go back to those families and look more carefully.